Disclaimer, this presentation represents my own research and opinion does not necessarily reflect the views of my employer, Revision Skincare. There are going to be 3 learning objectives today, so the goal is for you to walk away from this presentation with at least one thing that you've learned. The first objective is to accurately describe the term cosmeceutical, to understand clinical study terminology pertaining to cosmeceutical products, and the third is to understand how cosmeceutical companies use preclinical and clinical testing to prove product efficacy. And please, while I'm presenting, please go ahead and ask questions in the chat, or if you'd like to interrupt me, you can raise your hand and I can answer your question. So it can be very interactive. So let's go through the first learning objective. So if we research on PubMed the word cosmeceuticals, I did this earlier in the week, there were 1,254 results. As you can see in this picture here, or diagram, in 1996, there's about 0, and it has exponentially increased since then. And when I did this search 6 months ago, the first hit was an approach to cosmeceuticals, and the second hit was cosmeceuticals, what's real, what's not, interestingly enough. And then let's skip 4, 5, and 6, or 6, 7, and 8 were really interesting as well. Speaking to the cosmeceutical marketplace, it's the fastest growing sector in personal care cosmetics. Dr. Zoe Drelis is also going through this publication discussing what's real, what's not, what to believe in, what not to believe in. So let's deep dive into cosmeceuticals. Where does the term come from? It was first coined in 1961 and published by Raymond E. Reed, who was a founding member of the Society of Cosmetic Chemists. And he put together 2 words, cosmetics and pharmaceuticals, to designate an active cosmetic. It was recoined by Dr. Albert Kligman, MD, PhD from the University of Pennsylvania in 1984. And in his definition, in his publication, he described cosmeceuticals being scientifically designed products for intended external application to the human body. And the product has pharmaceutical activity, but it's more so it's intended for skin that is that's normal skin, near normal skin, so nothing disease state. And the product is to possess a very low risk profile. So when we ask what are cosmetics or cosmeceuticals, think of vitamin A, vitamin C, hyaluronic acid, ceramides, peptides, caffeine, and kojic acid. All right, that's definition from Dr. Kligman and Raymond E. Reed. What does the FDA think about cosmeceuticals? So here we are. This is on the FDA's website, and I'm just going to read the first few sentences. The term cosmeceutical has no meaning under the law. While the Federal Food, Drug, and Cosmetic Act does not recognize the term cosmeceutical, the cosmetic industry uses this word to refer to cosmetic products that have medicinal or drug-like benefits. To the FDA, a product is either a drug, a cosmetic, or it can be both. So a conundrum here. So we know that there's so many publications in the academics and in industry speaking to cosmeceuticals, but the FDA doesn't like to talk about cosmeceuticals, does not recognize this term, and as well as the European Union. Okay, so I'm going to give you some examples, and then we're going to go back to what the FDA tells us. So Bakuchiol has been such a highly researched ingredient. It's been an ingredient on the market that I think has increased attention and been very attractive to many consumers. And so here are 4 examples of Bakuchiol-based cosmeceuticals. And Bakuchiol is that ingredient that has been said to be an alternate to retinol. It comes from the bapshi plant. It has roots in Ayurvedic medicine, so traditional Chinese medicine. It has a very similar gene expression profile to retinol. So it's very attractive in this cosmeceutical industry. You have Herbivore, this company called the Inky List, and then you have Paula's Choice and this Bakuchiol Skin Restore. I was really interested in the clinical Paula's Choice, and I wanted to deep dive into that as when we're talking about cosmeceuticals, what is Paula's Choice in a clinical manner speaking to Bakuchiol, and where are the claims coming from? So if you go to paulaschoice.com and click on a video, the video takes you into explanation of this product. It contains both retinol and bakuchiol. The first three claims are fragrance-free, paraben-free, and cruelty-free. Furthermore, the first claim states, visibly reduce deep wrinkles, fine lines, dark spots, and discoloration. They performed a clinical study, they say. 94% agreed it's visibly improved elasticity, and 90% agreed in improved skin texture. So this was a consumer in-use study with 78 subjects after 8 weeks. So when we look at these claims, they're very compelling to the buying consumer or the purchasing consumer. 94% agreed that they visibly saw an improvement in elasticity. I mean, I would buy that if I actually, if I was a purchasing consumer. And when you really deep dive into their website and you look into where are their claims coming from. Now, retinol is well established. It's a well-established cosmeceutical. There's 20 years of history on retinol and its mechanism of action. Bakuchiol, maybe a few years of information, but when you look into their website, you see that their research, their claims are not because they perform robust clinical efficacy studies, but it's because they utilize research from peer-reviewed journals to substantiate their work. Not saying this is wrong and not saying it's right. This is just one method a company has taken to look into launching their cosmeceutical product and defending their claims. So we'll go into more of the good, better, best scenario at the end of the presentation and my thoughts on how to really clinically design for studies to really support your claim substantiation. So of course, I love Wikipedia. Who doesn't? I always want to see what Wikipedia is telling our consumers. And I underlined in red something very unique. There are no legal requirements to prove that these products live up to their claims. Kind of scary though, right? So we can launch products into the industry and they don't have to have any legal requirements. So that's one thing to ponder. So at least Paula's Choice has a consumer self-assessment questionnaire that rated how well a patient's skin was looking after 8 weeks. Some companies can launch products without any sort of legal any sort of claims because it's not legally required. So let's deep dive into that. So it's true, the FDA does the FDA have authority or approve before they go on market? So, and who's responsible for substantiating the safety of cosmetics? The FDA legal authority over cosmetics is different from authority over their other products we regulate, such as drugs, biologics, and medical devices. So they are specifically stating that they really do not have authority and approve cosmetics when they go into the marketplace. And who is responsible for substantiating the safety of cosmetics? Companies and individuals who manufacture or market cosmetics have legal responsibility to ensure the safety of their products. And so that's very unique. So it is up to the supplier, the manufacturer, and pretty much what their legal regulatory team decides on how to launch their products and substantiate their claims. So knowing all this, some of the key takeaways from this first section is that the word cosmeceutical combines cosmetics and pharmaceuticals to designate active cosmetics. The cosmeceutical industry is the fastest growing industry in personal care products. Although the term cosmeceutical is steeped in dermatology literature and dominates academic discussions, this category of skincare is not recognized by the FDA. And brands incorporating key active cosmeceutical ingredients can build compelling claims to help drive consumer acceptance and purchase. So I'm going to stop here to see if there's any questions. And I actually also believe that I'm using the not the most updated presentation I created, but it's okay. Are there any questions right now from the audience about cosmeceuticals?
I have a quick question.
Sure.
The definition, or might have been the Wikipedia, said there's no legal, that's it, no legal requirement to prove that the products live up to their claims. But don't claims in general need to be legally substantiated?
For cosmetics. Yeah, for cosmetics. Because if you're an OTC or a drug, there's more, the FDA definitely does that, has to control that. You'd have to submit more information to the FDA. And so really the FDA doesn't even require The FDA doesn't even require to review safety testing on our cosmetics, on the products being launched into the marketplace. So it's up to the brand and the supplier and the manufacturer to actually put together that dossier and have it internally.
I see.
And I do have my regulatory team on as well, and they're subject matter experts, but maybe they can possibly add in some more things into the chat if they would like to.
Yeah, I'll type something in the chat if you want, Alisar.
Thank you, Taylor. I appreciate it. Great, thank you for that question. All right. So the second objective of the presentation is to understand clinical study terminology pertaining to cosmeceutical products. So this might be a little boring in the beginning, but we're going to try to make it fun because it's a lot of words that are going to be thrown at you. So looking at clinical terminology, let's start with clinical trial study. So it is a research study in which one or more human subjects are prospectively assigned to one or more interventions of those interventions, which may include placebo or other control, to evaluate the effects of those interventions on health-related biomedical or behavioral outcome. And of course, this definition is coming from the National Institute of Health, NIH, and not really anything related to cosmetics per se. Preclinical study, it's performed in an in vitro, ex vivo, and in silico models to obtain basic information about the safety and biological efficacy of a drug candidate before testing in a final targeted population, such as humans. A consumer in-use study evaluates the performance of products and provides information on consumer behaviors in order to design a product whose use is easy and practical in consumers' lives. The evaluation of the product is performed by consumers prior to the product launch. Now we're going to go into really in a clinical trial, you can perform different types of trials. One can be an open label study, and this is the type of study in which both the health provider and the patient is aware of the drug treatment being given. And a double-blinded study is a type of study in which both the health provider and the patient are not aware of the drug or treatment being given. And typically you could have this in a split body or split face study in terms of dermatology, or you can have one cell being treated with the active and the one cell being treated with the placebo. So what is an active and what's a placebo? The active is a substance or treatment that's given to a human subject with a therapeutic effect or a hypothesized therapeutic effect. And a placebo is an inactive substance or treatment that has no therapeutic effect, although we know that placebos have a placebo effect and there may be an effect per se. So when we say placebo controlled in this type of study, especially when you look at publications, the key thing to look at, and we'll be discussing very soon, is the results obtained between the two groups. So when you have a placebo control group and an active group, you want to see that the active group outperforms the placebo. And that is why you run a placebo-controlled study, to ensure that the vehicle control or that placebo isn't really affecting or changing structure function of the skin, for example. A randomized study is a powerful tool that helps control bias in a clinical study. So you're randomly assigning a study participant's treatment of either the active or placebo or perhaps the comparator product. And the randomization key is performed prior to the start of the clinical trial, and it's pretty much locked and no one's allowed to really see the randomization key to keep both the study participants and the clinical grader blinded during the entire time of the study. My last definitions for you today is white paper, peer-reviewed manuscript, and poster publications. So a white paper is a marketing tool in the form of information on the technology of the underlying complex product and its system. The format is not reviewed by peers, which are considered subject matter experts or accomplished scholars within a given field. But there's always a place for these white papers as they quickly provide information to the HCPs, to the physicians if they need to review such documentation. Peer-reviewed manuscripts involve subjecting the author's scholarly work and research to the scrutiny of other experts in the field. So when we look at publications today from the Journal of Drugs and Dermatology or the JCAD or ASJ, every time you write a publication or manuscript, you submit it to the peer-reviewed process, and there are these experts that review and criticize your work positively or negatively and decide if they want to keep your publication or reject it. So they're pretty much providing information to the publisher to decide whether the work is to be accepted or not. And a poster publication is a scientific conference committee. They review this, the submitted poster abstract, and they evaluate for suitability for publication. So depending on the scientific conference you attend, there's always a process for submitting your abstract, getting that approved by the committee, and then submitting your poster and then getting that approved too. So it's definitely a more rigorous process on peer-reviewed manuscript and poster publication versus the white paper. All right, so when we look at different types of publications, here are some examples. So the first top example, I took it from my work site. I like this publication from the ASJ, a randomized double-blinded split body placebo-controlled clinical study to evaluate the efficacy and tolerability of a topical body firming moisturizer for upper arm rejuvenation. It's a mouthful, but let's just break it down. So this is a randomized study, meaning that randomly subjects are going to be given the placebo versus the active. It's double-blinded, meaning that the subjects are unaware which product is the active versus the placebo in a split body. So the subjects are using their own body, and they're going to be placing the product, the placebo and the active, on different parts of their body, and it's going to be split right and left. So that's an example that was recently published in the Aesthetic Surgery Journal. Here's another example, a randomized controlled trial of skincare protocols for facial resurfacing, lessons learned from plastic surgery, educational foundations, skin products assessment, and research study. So here's another example of a publication. And then this example, pore refiner shrinks pores, smooth skin. So this is Dr. Zanabaji, and this would be an example of a white paper. And the last example is another robust study. A high-potency, multi-mechanism skincare regimen provides significant anti-aging effects, results from a double-blind, vehicle-controlled clinical trial. So here's just some examples that you can see that are focused on the clinical work. Now, these examples are from preclinical studies. And so the top left, you'll see upregulation of extracellular matrix genes corroborates clinical efficacy of human fibroblast-derived growth factors and skin rejuvenation. So you see here, this is focusing on how to utilize different models to look at matrix genes that can be upregulated due to a treatment. And gene expression studies pertain to extracellular matrix integrity and remodeling, nuances and pitfalls of in vitro investigation. And the last example here is epidermal and dermal hallmarks of photoaging are prevented by treatment with nighttime serum containing melatonin, bakuchiol, ascorbyl tetraisopalmitate in vitro and ex vivo studies. So the key takeaways in this section is a clinical study involves testing an active product to determine efficacy in a controlled setting, in some cases versus a placebo, vehicle control, or comparator product. Preclinical studies using cell culture models or excised tissue to elucidate product mechanism of action, and a double-blind, and we'll go more into these preclinical studies in the next section. A double-blinded, randomized, placebo-controlled study is considered a highly robust clinical study method. I think someone put it in the chat, the gold standard. So yes, that is true. It's a very gold standard in dermatology or in clinical science. This type of study is typically found in medical devices and drug products. So you don't typically find a double-blinded placebo-controlled study, even a split body or split face in the cosmeceutical world. It's very much more for the medical devices and drugs. Doesn't mean it doesn't need to be there. I think that it really just sets those scientists and those companies apart from others. And then brands publish their clinical work in several formats, including white papers, peer-reviewed journals, and posters. So I'm going to pause here and see if there's any questions. And I hate to do this to you all, but I need to open up my newest presentation because I need to find that. So are there any questions while I find the correct presentation?
I have a question.
Yes.
You may have kind of already covered this. I just want to make sure I have it correct in my head. So for instance, a lot of suppliers have actives for anti-aging and things like that, and they've done their own clinical studies, and they have all this clinical data with the usage level that they used. So if a company or a contract manufacturer uses that particular active at the level that they use in their clinical study, can they still make those claims, or do they need to, is the standard that they would still need to go have their own clinical study done on their finished product with that active in it?
So that's a great question. And that's the model that I was going to talk about at the end, like good, better, and best. When we look at adding in any cosmeceutical ingredient into a finished product, we tend to look at adding at the right concentration, because in that concentration, you're supposed to see an effect, right? But the right methodology, if you have, and it really just depends on the size of the company, the amount of, dollars the company has for the clinical science. The best thing you would do, Chrissy, is to run a study on your finished product, because you need to substantiate the full product and not the individual. The individual ingredients are important. They make the entire story, but the best thing you can do is run the study on the full product itself. So
Okay.
it's not a black or white. It's very in between.
Right.
Right.
Okay.
But great question. I really hope this is the right presentation. I was adding a few more things before we started talking today, but I love the engagement. Any more questions? That'd be great. So this is the last objective, and then I'm gonna and it'll take some time to go through it, because what I want to do is give you examples from publications and talk to you about how these scientists write a publication, look at the type of science is being brought into the industry, and let's just critique it together, positively or negatively. A critique is a critique. So preclinical studies. These are in vitro models that include single cell culture or full thickness tissue. Ex vivo models include skin excise from plastic surgery. And designing in vitro and ex vivo studies can really, if you have never designed a study before, to Chrissy's point, she stated that there are several suppliers that demonstrate and explain the mechanism of action of each of their featured ingredient. And there are several companies that do it really well. Sederma does a great job looking into every ingredient and the mechanism of action. So these are companies that really spend the dollars to figure out the mechanism of action. And that helps us scientists, clinical scientists especially, to understand if those ingredients should be placed into the product. So we could utilize the templates, the method from the supplier, collaborate with CROs to develop similar in vitro and ex vivo models to test finished product efficacy. So these CROs are all over the globe in our country that you can Google and find, and you can work with them, basically take the information from the ingredient supplier and change it a bit to fit your finished product. Also working with established working relationships with universities. There are several universities across the country that are focused on pharmaceutical cosmeceutical sciences, and those universities always look to have funding from corporate America. And then finally, review, publish preclinical data from industry experts to gather ideas on how to design preclinical studies. And I think the last one would be really network. Go onto LinkedIn and meet some of the scientists and ask them for help. I mean, I'm sure they're willing to help anybody in developing studies. So when I want to deep dive into some clinicals, and I hope you guys can see this because it's very small on my screen, but I'm hoping that you can see this pretty well. So the next few slides, we're going to look at the different clinicals and just examples. So this clinical was published in the Journal of Drugs and Dermatology in 2019. And the materials and methods section of the JDD discusses utilizing the MatTek epiderm full thickness model. So this is an in vitro full thickness model, and they're specifically looking, this is the company called Alisar, and they make it known here, Alisar Skincare Regenerating Skin Nectar, and they're looking at gene expression. It may be very small for you, but they're looking to see across the different genes of the dermal proteins like collagen and elastin, is there any upregulation? And based on the data from this preclinical finding, what are the claims they can establish? So they say that because they saw statistically significant upregulation of matrix genes after 72 hours, then they can claim that their product, Elastin Skin Nectar, remodels the dermis. So this is preclinical work using full thickness in vitro models. Here's another company, Skin Better, published at Scale, and they looked at they have an antioxidant product, and they looked at their product in an in vitro model and how well its antioxidant package can quench or help prevent free radical formation. So what you can see here on the y-axis is the measurement of blue light, and on the x-axis, different degrees of reduction of free radical production due to either cigarette smoke, for example, what they did here, or blue light. The one thing you'll notice between these two figures is the way that the figures are put together. It is utmost importance for us scientists to really establish really great work and really develop figures that are very easy to read and to replicate in a presentation. So just as the one thing to know, and when we do figures, is something similar to this is definitely not optimal, but a figure like this is more optimal because you can clearly understand without having to read the subtitles here. So that's just an example on how to develop preclinicals, but also how to design them in a figure format that can be easily translated and understood by us scientists. I wanted to look at another ex vivo model. So the first two were in vitro. These are ex vivo models. Here's a company called Isden, and they've performed an ex vivo model to look at if their product can help prevent sunburn cells in an ex vivo model. So this is their anti-aging treatment. All the details are here on how they actually ran their clinical study in the ex vivo format and how much they irradiated the skin. And then they measured on the y-axis sunburn cells versus the different types of treatments. And they show and explain, moreover, our data suggests that the combination of melatonin, Bakuchiol, and ATIP together exerts a synergistic effect on skin biology and reversing signs of aging acquired as a result of UV exposure. So they're making claims on their product based on this ex vivo study. So now you're starting to see the pattern. There are preclinical studies that are performed by companies on their finished product to understand what the behavior is. So we first looked at in vitro upregulation of genes and then looked at the finished product and how well it can mediate free radical production and reduce free radical production. Here's an ex vivo example of skin biology and prevention of sunburn cells. So those were the preclinical. So in clinical testing, we're going to talk about subjective grading and objective grading. I know I have 15 minutes, but I think I can get it done with more discussion with you all. So when we look at clinical efficacy grading, it's conducted by highly experienced and trained clinical investigators. So typically physicians and PhDs. The assessment is using a validated scale. There's a Griffith's 10-point scale. It's a photonumeric scale developed by Dr. Christopher Griffith, and a global aesthetic improvement scale called the GEIS score. It's a 5-point scale that also is used predominantly in dermatology. Of course, when you're also creating a clinical efficacy study, you have to look at tolerability and safety. So some of the measurements that are looked at are erythema, edema, and peeling. And these tolerance scales are very simple. 0 is none, 1 is mild, 2 is moderate, and 3 is severe. So this is the investigator reviewing someone's face and deciding how well the treatment is and helping prevent erythema, edema, and peeling. So this is Dr. Christopher Griffith created this in the scale in 1992 and published the work, photonumeric scale for the assessment of cutaneous photo damage. And so this is just an example of where these scales come from. So it takes several years of research and taking many different pictures of subjects across several years to look at what the photonumeric scale means. So if you look at the bottom here, the rating, 0 is none, 1 to 3 is mild, 4 to 6 is moderate, and 7 to 9 is severe. So if you look at this publication, what he has done is every single grade has a picture to it, so that when a physician is grading based on photo damage, he or she can look at those pictures and then grade accordingly based on the criteria that was developed by Dr. Griffith. So these are examples of clinical efficacy grading, and it's also known as subjective grading. So let's deep dive into a publication on how a subjective grading was used. So this was published in the JAD, and you can see the clinical efficacy was looking at overall photo damage, global face, under eyes, wrinkles, and there's more information here. Efficacy parameters are listed below. Oh, I kind of deleted that part. But in the figure and the results on the top right, you'll see that this is how this company has, the scientist, Dr. Rahul Mehta, has shown his science, and it's actually very well and easily read. If you zoom in, you can see it very well. On the y-axis, you have the percent of subject improvement. On the x-axis, you have the time and the time of treatment. And you can see that there's a decrease over time, which means there's an improvement. And of course, you have clinical photography showing before and afters to show the user, the reader, the consumer, the patient, the doctor, that this product can improve signs of aging. So that's just an example of subjective grading. Objective grading by bio-instrumentation minimizes subjective grading bias and ensures repeatability by minimizing result and variation. For the ones that cannot see the slide deck, sometimes you have to sign out and sign back in to Teams. Teams is sometimes odd like that. So when we think about bio-instrumentation, it's very different because it does reduce bias. And examples of those instruments are cutometer, which measures skin elasticity, corneometer, measuring skin hydration, ultrasound, measuring skin density and skin thickness, and then Vizia. Vizia is a platform that I think everybody may have heard of. You pretty much put your face in this big camera system, and it takes many types of pictures under different filters. And we're going to give you some examples of those. And Vizia CR has a really robust tool that can also measure wrinkles, pore size, photo damage, brown spots, dark spots, you name it. That the company Canfield has a really robust method of visual measuring different types of analysis on the face. So this publication comes, this publication we talked about before is that it is a high potency multi-mechanism skincare regimen. It's a double-blind wrinkle-controlled clinical trial. But what I wanted to show you is figure 6. So this is an ultrasound image of an increased dermal density over 30 weeks demonstrated by the ultrasound imaging in the subject. So what it's showing at week 0, week 8, and week 30. So to orient yourselves, the top section, the left part, is the epidermis, and below is the dermis. And what it's showing you is that after week 8 and week 30, there's an increase and improved echogenicity. So there's more dermal tissue, there's more dermal proteins, within the dermis. So when you see more green, there's an increased echogenicity, meaning that the product itself is changing structure function of the skin. And what they've done here is statistically show that the active statistically improved versus the placebo, or the vehicle. Very nice data, really showing that you have to compare your active versus your placebo. Here's some clinical photography from Vizia using standard light. So standard light doesn't really provide details it provides more surface details and not underneath the skin. There's different ways and different methods to look at that as well in Vizia, but this is just an example from the JCAD journal, where you have A, B, C, D is different time points on the subject and looking to see improvement in under eye fine lines and crow's feet wrinkles. So an example of how Vizia and clinical photography really helps you understand how products are efficaciously improving skin health and skin features. And this is a before and after of a subject utilizing a product. This was presented at the AAD in 2019. But one thing to notice, this is Vizia CR captured under cross-polarized light. So when you look at cross-polarized light in Vizia, it's providing features underneath the skin and really pulling up those vascular features, very different than standard light. Another type of filter that also Vizia has is parallel polarized. So really Vizia is set up for taking really nice before and after pictures. Here's an example of not the face, but the body from Elastin. They're showing a bland moisturizer versus Elastin's treatment, and just another type of way to show consumers and physicians the improvement of Elastin versus their product without quantitative analysis, but more showing before and afters can say a lot of things there too. Okay. When we look at self-assessment questionnaires, SAQs, these are consumer perception questionnaires, and they're unique because they typically help the marketing and brand team create claims. You saw Paula's Choice, right? That's an example. We'll give you more examples in the next slide too. There are questions that are asked at baseline, during the study, and at the endpoint. I believe this is the entry point into cosmeceutical market. I think that it's an important thing to have. It's an entry point. It's perceived well by consumers, especially those purchasing consumers. And it's not a replacement, though, for robust clinical studies. So here's an example from Skin Medica and talking about how many weeks younger the skin looked across a broad patient population that was studied. And then this is coming from the Derm Booster Bakuchiol Better Anti-Aging, or from Beekman 1802, a company that you can find on Alta MD, or also on, I think, Derm Store. So 95%, 95%, and 100%. And these are all based on consumer studies, so consumer acceptance studies. Another publication in the Journal of Cosmetic Dermatology coming from 2020, and it's a little bit hard to see here, but this is all these questions were asked during the study, and it tells the percent of subjects that actually agreed on that statement. So the key takeaways from this section is that preclinical studies assist in elucidating the mechanism of action of a cosmeceutical. Some ways of elucidating the mechanism of action is utilizing in vitro models, cell culture models, and looking at gene expression, protein expression, or even looking at claims of cellular protection by antioxidants. And studies demonstrating this, some of the claims can be used for claim substantiation. Objective clinical grading using validated photonumeric grading scale is a gold standard. In dermatology, however, there's some bias. And to reduce some of that bias, objective grading using bioinstrumentation can be implemented and offers an alternative approach. And that can be providing claims like moisturization claims, improvement in skin elasticity, and improvement in skin density. Data gathered from the SAQs and consumer perception really provide entry-level claims, helping into the market space, and they really just drive consumer acceptance. So my closing thoughts in the last 5 minutes of the talk on new product cosmeceutical commercialization. So when we look at developing cosmeceutical products or new products and bring them into the market, there is always a standard. There's always a standard or industry standard that we have to meet. So I call this good. So we have to, I think that it's really based on morals and ethics, we have to ensure that our product is safety tested. So typically that's by human repeat insult patch testing. Also, we have to make sure that the preservative system is robust, so there's not going to be any bugs growing in the product, and so that we can reduce any incidence of issues by the consumer. So that's microbial testing, and product stability or packaging compatibility, ensuring that although OTCs only require a shelf life, cosmeceuticals and cosmetics should as well, just making sure that the package is compatible with the product. What's better? The next level. You take the good, you add a consumer perception and clinical photography based using a case study. So using based on case studies. So not a full clinical study, but maybe a small case study that can be performed. And what's the best, like the highest in my mind, and what I've seen across the industry is taking the better, which incorporates the good, adding preclinical studies, adding controlled clinical studies, and bioinstrumentation. So this is like what I would say be the gold standard in submitting a new product into the marketplace. And so in conclusion, the word cosmeceutical is currently not regulated by the FDA. Although cosmeceuticals span active ingredients which have been shown to influence structure, function, skin properties, it is again not approved or regulated by the FDA. And this is a very fast growing industry in personal care cosmetics. So I would say all of us have some nice job security in that. And designing clinical studies to support cosmeceutical product claims requires a multimodal approach. So preclinical testing, in vivo testing, utilizing clinical efficacy, grading, bioinstrumentation, and self-assessment questionnaires. My last thought is that each brand has their own requirements for launching new products, and it's really their decision to determine the best approach and weigh the legal risks they are willing to take. And that really is up to their legal team and their quality regulatory team, mostly the legal and regulatory, to decide on what that approach is going to be. And so with that, I want to thank you all for attending. I want to acknowledge the Southwest Chapter, Society of Cosmetic Chemists, for inviting me, my mentor, Tatiana Kononov, my team, Sophia and Morgan, and my regulatory partner, Taylor, who is honestly, she's a whiz at everything, and she provided some input to some of your questions on the regulatory. So thank you so much for your attention.